Presenting the first Australian record of Anatoxin-a production by Microcoleus. Microcoleus autumnalis is a well-known anatoxin-producing benthic species in New Zealand and the US. Last year, a new species, Microcoleus anatoxicus, was described from Northern California. Dog deaths have been linked to Microcoleus, and there is little known regarding the impact on wildlife and stock.
Relevant previous research.
In South Australia, an investigation into benthic formidable growths were observed to be toxic by mouse bioassay. Anatoxin, however, was not detected. Genetic PCR assays targeting the anatoxin biosynthesis genes usually test for the specific genes ANA-C or ANA-F as illustrated in the schematic representation here. In a 2020 paper, sampling methods and the detection of a number of genes for Benthic Cyanobacteria within reservoirs was presented. Whilst detecting the ANA-F gene in 2 samples, the identity of the cyanobacteria was not established. A 5-year study in Victoria testing surface water samples detected the ANA-C gene from nested PCR in 31 of 67 sampling locations. Anatoxin production was confirmed using an ELISA kit. However, the actual species responsible, or whether they were planktonic or benthic in origin, was not established. Discovered during a weekend family walk, Dungay Creek within the Tweed LGA is the site for the first record of Australian Microcoleus positive for anatoxin. More weekend forays followed, and 8 localities throughout the Tweed Valley catchment have now been shown to have Microcoleus mats that have the capacity to produce anatoxin. Microscopically, the species of the Microcoleus formidable complex are virtually impossible to distinguish. Microcoleus is characterized by having several trichomes within a common sheath. Cells are generally isodiametric and attenuated at the end of filaments. Using ANA-C positive controls from a New Zealand Microcoleus and an Australian Cuspidothrix, mat material from several sites across the Tweed Valley catchment were tested using the following: Microcoleus autumnalis specific assay, a non-specific ANA-C assay used in the Victorian surface water study without the initial antigen and amplification primers, phytoxygene cyanodetect assay, a melt curve analysis following amplification using the 4 ANA-C assay showed no perceivable difference between the positive New Zealand strain and all 8 environmental extractions, suggesting that the mat material is either Microcoleus autumnalis or a closely related species. When using the non-specific NSC primers, a melt curve analysis again showed no difference between the 8 Tweed environmental samples and the New Zealand strain of Microcoleus, but not surprisingly do differ to that of the cuspidothrix strain. Glenn McGregor microscopically identified the Tweed mat material as Microcoleus. He and his team also were able to perform single filament isolation for culturing. Barb Sindel was able to verify the PCR with a different non-specific NSC assay. Additionally, using LCMS, Queensland Forensic Scientific Services were also able to detect Anatoxin-a from mat material from the Dungay Creek environmental sample. Shown here are the water quality parameters when mat formation was maturing at Dungay Creek. The riffle upstream is contributing to the high dissolved oxygen level recorded. The New Zealand streams, phosphorus levels below 0.05 mg per litre have been found to be favourable to the growth of Microcoleus autumnalis over other periphyton. Mat formation starts with filaments of Microcoleus colonizing substrate, which may already have sediment on it, but the mat also traps sediment over time. The length of time undisturbed during mat accrual will be important in determining how thick the mat gets, and therefore the potential toxin load. Growth can be very quick, as shown in a field-based experiment conducted in Canterbury, New Zealand, which in just 16 days recorded a 200-fold increase in Microcoleus biomass. Microcoleus growths have been observed in Tweed on cobblestones, but also found on rock walls and concrete causeways. Mats in Tweed can be almost purely composed of Microcoleus, but more often is associated with a range of other periphyton, most commonly filamentous green algae, diatoms, and also a variety of other cyanobacteria. The New Zealand experience has shown that even when Microcoleus isn't the dominant algae, Anatoxin levels can still be a significant risk. In New Zealand, water temperatures above 15 degrees Celsius is considered to be an important factor encouraging growth of Microcoleus, and at a representative site at Ukai and Tweed, it is well above 15 degrees for most of the year. Water flow is another key factor in the overall cycling of Microcoleus mats. Mean rainfall over the year shows a fairly predictable pattern of high summer rainfall through into autumn, then lower rainfall through to late spring. The Tweed Shire LGA covers 1,300 square kilometres, with Mount Wollumbin the centrepiece of the caldera. The observed NSC positive Microcoleus sites are situated to the western upper reaches of streams and the main Tweed River. East of Murwillumbah, the Tweed River is estuarine. Positive mats were found upstream of all 3 drinking water supplies. So what is the risk posed by Microcoleus proliferations? In the reaches of streams and rivers where mat proliferations occur, the most obvious risk is when dogs, livestock and wildlife eat the Microcoleus material. Toxin release through leakage and mat detachment has been reported downstream and therefore may be a concern for water authorities. As mentioned, Microcoleus is known from several states within Australia. It's just not routinely tested for the capacity to produce anatoxin. In New Zealand, it is recorded from over 100 rivers throughout the North and the South Islands. The increase in the prevalence of proliferations in New Zealand is thought to be largely driven by changes to riparian margins and increasing nitrate and fine sediment loads. It is time to get your waders out and start looking for Microcoleus in Australian streams or rivers near you.